Am I audible? Is the screen visible? Cool. Okay, so this is the second tutorial or the tutorial for the second week of I Think Biology, which is a course offered by Azim Premji University. I'm Gitika Agarwal. I'm a PhD scholar at the Center for Ecological Sciences, and I'll be taking this tutorial for you guys today. So, in this week, the major topics that were covered includes they talked about numbers and scales in biology, how different things are measured in biology, and estimation and allometry. We'll come to what allometry is later in the slides. Then they talked about how experimentation differs from theory and how theory feeds into experimentation and vice versa. They also discussed finding reliable information for purpose of research or for the purpose of writing an article, and reading a scientific article or how to better read a scientific article and understand it. So, moving on to the first question, what is quantification? So, basically, what quantification is in the case of science is that it is a practice of counting and measuring parameters or variables. So, when you start out with research, in general, in science, it is central. Quantification is a central practice because you need to uh, go on and come up with a proof of what you have done and how it is accurate and statistically significant. So for that, quantification is a very important practice, and it basically involves counting or measuring different parameters or variables. For example, if you're working with for example, say trees, and you want to find out how many trees are there in a particular landscape, you need to go on transects and count the number of trees that you encounter of that particular species, and that will help you estimate the total tree count in a particular area. So, another thing to keep in mind is that measurement is useful only when you know what you want to measure. So, it is very important to determine beforehand that what you want to measure. Only then can you start quantifying it. So, therefore, quantification must always follow a qualitative description of the system. So, by using quantification, you cannot come up with a model. So, because you need to first have that model, or you need to first understand your system before you start carrying out quantification. Okay. So, another thing is quantification. As I said, it cannot be used to generate the model, but it can only feed into your model and it can only refine your model further. So, if we discuss of what all things can be quantified in biology, so I have made a list of few things that you can quantify in biology. One is, say, cell counts. If you're working with a bacterial colony, or say you're working with Escherichia coli, so you can carry out a cell count or a colony count in that matter in your petri dish. You can count the number of mutations that have happened from the ancestors to the newer generations. You can look at population demographics. For example, what is the number of males in the population? What is the number of females? What is the number of people aged above? If you're working with humans, for example, you can look at people aged above 50, people between 25 to 50, and people below 25. So this is what basically is population demographics. Then you can quantify body size or body mass of different organisms that you might be working with. You can look at the rate of metabolism. If you're working with, say, the anatomy of something, you can look at how what is the rate of metabolism in that particular organism or the species that you're studying. And you can also look at, for example, things like heart rate. If somebody is a medical practitioner, it might be important to them to look at the heart rate of their patients, and many more things in biology. So, can you think of a few things that can be quantified in biology? You can type it out in the chat box and let me know if you know about a few things that can be quantified in biology. Okay, so moving on. So, how do you go about quantifying variables, or do rates count as quantifiable parameters? Yeah, if you're looking at, for example, you're talking about the rate of metabolism, right? So, but if you're looking at metabolism and you're looking at what rate metabolism is happening in a particular uh, organism or in a particular species, that is quantifiable. Anything that you can, for example, represent in numbers is a quantifiable parameter. Okay. So, yeah. So, here is a sport that you might be very well aware of. So, how will you quantify, for example, or what things can you quantify when it comes to cricket? It's just a simple exercise. So, what all things can you think? Yeah, the number of runs the cricketer scores. You can look at the number of wickets the bowlers take, and other things that are quantified are like what is the speed at which the bowler threw the ball, um, what is the run rate that the cricketer is scoring. These are a few things, or so like that can be quantified in the sport of cricket. Then, um, in this image, which is of a bird singing, what do you think can you quantify when it comes to looking at the vocalizations of something like a bird? Yeah, you can look at the loudness or the amplitude at which it is vocalizing. You can quantify what frequency it is vocalizing at. You can also quantify the time period for which it is vocalizing without a break. So these are some things that you can quantify. And let's move forward. Okay, another thing. So if you are um, given a pain scale, for example, doctors mostly ask their patients what is the level at which they're feeling pain, which might range from no pain to severe or very severe pain. This is also a form of quantification, because mostly in psychology or in the terms of pain, you cannot actually assign a particular number or an exact number to the amount of pain you're feeling. So that is why the researchers mostly give different classifications, and they assign an arbitrary score to those the pain levels, and that is how this pain scale works. So all these levels are not like some accurate measurement, they don't have like a unit, but still they are referred to as quantifiable variables, because we are trying to find out how much pain the patient is going through. Okay. So there are two forms of scales in which biological parameters that can be used in biology. One is the spatial scale and one is the temporal scale. So as you might already know, spatial scale mostly refers to measuring the change in a variable across space. So when you move, say, from one geographical region to another and you measure a similar variable, then that is a spatial scale of measurement, or you're measuring change across space over some area. So, for example, there are different scales at which this can happen, or different levels. One, you could be studying a single cell, which would be a microscopic scale. You could be studying a forest patch. At a local level, you could be studying an entire ecosystem, which would be a regional level, or the entire planet, which is like a global scale. And why is the spatial scale important? That is because biological entities follow a hierarchy of organization. So here you can see there are cells. This is for a plant and this is for a human being. There are cells. These cells come together and they form tissues. Tissues assemble and form organs. Organs form organ systems, and the organ systems form organisms. So there is a hierarchy in biological organization, and at each of these spatial levels, you need to have a way to measure. So, for example, you could be measuring what is the size of these cells, what is the size of the tissue, things like that. So that is why it's important to keep in mind the spatial scale. And this is like some of the ballpark measurements of some things. So here you can see that, one second. Yeah. Here you can see, first of all, there is an atom. Um, that's the basic unit of any physical entity. And that comes around zero point 0.1 nanometers. If you move on to proteins or lipids, they fall between 1 to 10 nanometers, or more roughly 1 to 30 nanometers. If you go on to viruses, this is the size of mostly like the biggest virus, which is like 100 nanometers, but viruses fall in the range of 10 to 100 nanometers. Then viruses, moving on, there is the size of bacteria and mitochondria, which are in the scale of micrometers. Okay, let me take a step back and just tell you what these different symbols mean. So you might already be aware of what is a meter and what is a centimeter. But if you divide a centimeter into 10 more parts, that's called a millimeter. And a micrometer is actually 10 to the power minus 6 of a meter. Then if you look at nanometer, that is 10 to the power minus 9 of a meter. Okay, so this is to introduce you to these different mm, terms or these different symbols that are used here. So I was telling you that viruses mostly fall in the range of 10 to 100 nanometers. And if you look at bacteria or mitochondria, they follow in micrometers and not nanometers. Then from mitochondria, when you move on, or and an important thing to keep in mind is that the mitochondria is roughly the same size of a bacteria. And it brings out an important point because you might be aware of how prokaryotic cells like bacteria have transformed into or eventually, and there are eukaryotic cells. So it is believed or it is known that this was formed by the when one prokaryotic cell it ingested another one, and that is why, and all these so though the ingested prokaryotic cell became like organelles in that eukaryotic cell. So that is why the mitochondria, which is an important organelle which for metabolism in eukaryotic cells, it's the similar size like that of a bacteria. So this might be a question that might come up in the quizzes or the exams, and it's important to keep in mind how you can relate why bacteria and mitochondria have a similar size. Moving on to animal cells and plant cells, they follow in the range of a micrometer to 10 micrometers. Human eggs, which is like the largest human cell in the body, is about 100 micrometers. And now are sizes that you might be more aware of. There are like frog eggs, which are around 1 millimeters in length. Moving on, there are mice, which are about a centimeter, 10 centimeters, humans about 1 meters, houses and ships, just for knowing. So, like, just for bringing to your awareness that a house is like almost 10 meters in height, and humans are like 1 meters, and then there are ships which are like 100 meters in their length. Okay. Moving on to the temporal scales. So, just like there is a spatial scale, there is also a temporal scale of measurement in which you're mostly measuring change in a variable over time. It's essentially the time dimension of investigation. And you might be looking at the duration of a particular process if you're studying, say, uh, a particular process. For example, the Krebs cycle, you might be looking at how much time that takes to complete from the beginning to the end, or like looking at the digestion of food from when a person has consumed it to when it enters the small intestines. That could be like a duration of time, and you could be looking at time intervals as well between two such processes. So there is a different levels of temporal scales, beginning with the smallest one, which is like the molecular level, in which the processes happen at the nanosecond time scales, then subcellular and cellular levels, mostly microseconds and milliseconds. Moving on to tissues, organs, and organ systems, most of the processes happen at the level of seconds and minutes. Organisms, individuals, population, and ecosystems, the processes happening at these scales or at these levels span days, years, or centuries. Okay, so here is a figure to show you how different processes have different time scales. For example, if you look at reflexes, this is like a human reflex of somebody touching a cactus and then very quickly withdrawing their hand because of it being prickly. So reflexes happen in a time scales of microseconds to milliseconds. Heartbeats, there is like one heartbeats happen in the level of seconds because there are like 72 heartbeats per minute, so around seconds. Then the circadian rhythm that we are aware of, our 24-hour cycle. Ovulation cycle in humans spans 28 days, human gestation spans 9 months, and a human lifespan can span around 100 years. Okay, this is another diagram which is relating the levels of space and time. So when you look at biochemistry, when you're looking at molecules, they span around one, like in the domain of seconds when you're studying biochemistry. When you move on to physiology, you're looking at processes that span hours or weeks. Ecology studies years or centuries. For example, when you're looking at survival rates of a particular population, you might be studying that population over years. And if you're looking at evolution, that can capture processes or natural selection that has been happening for over millions of years. And similar goes for the space. If you're looking at molecules, as I said, they cover micrometers, cells span millimeters, and moving on to tissues, individuals are in meters, and populations, communities, and ecosystems might cover huge areas, more than a billion kilometers. Okay, so moving on to different levels of measurement. So you could be measuring things at a small level, as I already discussed, when you're looking at individual organisms. And if you're looking at, for example, their movement during their own particular lifetimes, how much they disperse, that is like a small scale measurement. If you're looking at immigration and emigration occurring between and among populations, that's like an intermediate scale of measurement. And if you're looking at geographical range shifts in response to, say, climate change or, say, tectonic plate movements, that is a large scale of measurement, both in sense of spatial scales and in sense of temporal scales. Okay, so there is this website which was mentioned during the lectures, which is called Bio Numbers. So I will just show you this website. So this is a website called Bio Numbers, where you can actually ask them or enter a lot anything you want to know. For example, if you want to know number of hair on human scalp or average number of hair, it will give you certain values. It can tell you what is the diameter of the scalp hair. It's around 60 to 80 micrometers. And it can tell you many other informations. So this bio numbers is a database of all of the biological numbers that have been recorded in research. So you can enter anything to get a rough idea of how much that size might be. For example, if you look at size of a, for example, say Escherichia coli, then it will tell you, uh, it is taking you to a table that tells you what is the some qualities about Escherichia coli, how prevalent it is in humans, and things like that. So this is a useful website that can be used, and it also has this key bio numbers uh, PDF with it, which gives you which marks down some key numbers that are used by cell biologists. For example, here you can see, one second. It's giving you the cell size of a bacterial cell, yeast cell, a mammalian cell volume. It's telling you some of the scales inside the cells. So, what are the size of the different organelles? Then it can be telling you what are the rates of division, of replication, or transcription and translation of DNA and RNA. Tells you different concentration, energetics. These are some important numbers. So you can refer to this website if you want to know something about these different biological numbers, or like these different measurements, and you can look up certain numbers in your free time or whenever you want to. Moving on to how there are two different scales again, now not spatial or temporal, but now looking at the measurement, linear or the log scale. So if you're looking at how are they different from each other, how is the linear scale different from the log scale of measurement? So when you're looking at the linear scale, it is an additive scale in which, um, when you move on in the scale, so however many points you move on, you add that many points to the number. For example, here you can say if you are at zero and, sorry, one second. If you are at zero and this is 100, so there are 10 equal partitions in between. So all of these numbers in between will be 10, 20. It'll be 10, 20, 30, 40, 50, so on. So it is equally divided into those parts, and it's an additive scale. But one of the cons is that it cannot cover a wide range of data. But when it comes to log scales, those are multiplicative. For example, here is a logarithmic scale. 10 to the power 0, as you might know, is 1. And when you move on to the next level, it is a multiplication of 1. This is 1 into 10. This is 10. This is 1 in this is 10 into 10, 100, for example. Similarly, this is 1000. This is 10 to the power 4. So it's a multiplicative scale. As you move on, you do not get like 1 plus 10 plus 10. It's 1 multiplied by 10 multiplied by 10, and so on. So to read this scale, so here you are at 10, and then you are 1 and 2 points away from it. So this is 2 into 10. Okay. So this will be 20. So this is how you read, how reading a logarithmic scale is different from reading a linear scale. In linear scale, you basically just add the values, and in logarithmic scale, you will have to multiply them. So a logarithmic scale, it can compress large values and allow a large range to be covered without losing the resolution for the smaller values. So this is how a logarithmic scale is important while measuring things in biology. As we already discussed, biology has hierarchical organization. So when you're talking about cells, and as we looked at that size from cells to human beings, we span from micrometers all the way to meters when we come to organisms as a whole. So you span from cells being in micrometers to them forming tissues, to organs, organ systems in the entire organism. So you span a large range of values, and that is why you might require a logarithmic scale, because it can depict all these large values, unlike the linear scale. Coming on to how do you read these graphs? So you might be aware of how to read a uh, linear graph, because both of these scale x-axis and y-axis are scaling linearly. It's pretty simple to read that. But when you come to these logarithmic graphs or semi-log graphs, in which one of the scales, for example, here they're looking at number of cells. So how are cells multiplying, or the growth in number of cells with time. So you can see when you are the number of cells are spanning a large range from 0 to 1,000, and the time scale is not going that much. That is why in this area you feel like all of these points are close to 0 only. But if you now convert this into a log scale, if you convert the y-axis into a log scale, you get a linear relationship between the two, and it's a lot easier to read this. And if you might be aware, the equation for for a linear line is y is equal to mx plus c. And here you just edit that, because here you have a log on the y-axis. So it's log y is equal to um, gamma x plus log a of lambda. This is the intercept, log of lambda to the base a, and x is the value on the x-axis, and gamma is basically the slope. So this is how the equation looks like: y is equal to lambda into a to the power gamma x. So this is how you interpret logarithmic graphs as, or semi-log graphs. If it's a if it's a log-log graph, then both of these axes will have a log scale, which we might come across later in the slides. And this one is a semi-log graph in which only the y-axis is on the log scale. Moving on to different scales of measurement. What are the different ways in which you can measure things? First scale of measurement is called nominal, in which you basically just classify things into different categories. For example, classification of organisms into kingdom, phylum, tax, these different taxa: kingdom, phylum, family, class, order. That is like a nominal variable. Then there is something called ordinal variables. In ordinal variables, you do have these categories, plus all of these objects follow a certain order. Again, giving you an example of kingdom, phylum. So that follows a particular order. You first have a species, then you have a genus, then you have order, and things like that. So that follows a particular order. In named variables, it might, for example, it might just be male and female. They don't need to be in a particular order. But for the ordinal scale, these values need to also follow a particular order. Then you look at the interval scale, in which they have the things are ordered, they have particular classifications, they there are proportionate intervals between them, but the zero on the scale is arbitrary. For example, the temperature scale, the Celsius and the Fahrenheit scale, both of them have an arbitrary zero, unlike the Kelvin scale, which is like the true zero. In Celsius and Fahrenheit, both of them have equal uh, intervals between two values, but the zero is arbitrary. And the last scale is the ratio scale, in which all of this is true, but it also has a true zero. For example, when you're measuring lengths, when you're talking about kilometers, meters, centimeters, that has an absolute zero. So that is a ratio scale. So these are the different kinds of variables, or like different scales of measurement. Now, this is a short video talking about Henrietta Lacks. This was mentioned during the first lecture, and it was mentioned that you should look into Henrietta Lacks and how her cells have played an important role in medicine and in research. So I have put this short video for you guys so that you can look at who was Henrietta Lacks and how what is the HeLa cells. One second, I will. Why is the volume not coming from there? Okay. I should share the. Oh, you're telling me to present the tab and not the entire window. Meet. How is it?
Henrietta Lacks was a sort of poor black tobacco farmer who grew up in Southern Virginia. And in 1951, she was diagnosed with cervical cancer. At that point, she had moved up to Baltimore, and she went to Johns Hopkins for treatment because it was the only place anywhere near her that treated black patients. So this was the era of segregation, and the hospitals had the quote unquote colored wards, which was the only place that black patients or poor patients could be seen. And without telling her, before treating her tumor, her doctor just cut a little piece and put it in a dish. And he sent that down the hall to George Guy, who was the head of tissue culture research at Hopkins. And George Guy had been trying to grow human cells outside of the body for decades, and it had never worked. And for reasons that remained a mystery, Henrietta's cells just never died. So they started doubling their numbers every 24 hours. They went from like one dish to two to four to eight to 16, and then just sort of pretty quickly took over the lab. And George Guy started calling his colleagues and saying, I think I have the first immortal human cell line, which is what they're called, which means they'll just grow and divide forever. And in response, his colleagues all said, great, can we have some? Because there'd been this enormous effort to grow cells outside the body for 100 years, because we really didn't know a lot about cells at that point. And in response to all of his colleagues asking for them, George Guy sent them to anyone who wanted to use them for research. And they spread around the world this way really fast. And they were one of the most important things that happened to medicine. They were used to help create the polio vaccine. They went up in the first space missions to see what would happen to human cells in zero gravity. They were used to create our most important cancer medications like vincristine and tamoxifen. Her cells were the first ever cloned, her genes some of the first ever mapped. They were used to help develop in vitro fertilization. Basically, almost all of the vaccines we take today can be traced back to research with her cells. There isn't a person out there who hasn't benefited from research on her cells.
The surprising tenacity of her cancer cells, which made them so important to medical research, also ultimately took her life. Henrietta Lacks died in October 1951, at the age of 31. As her cells were taken without her knowledge, in an era before the concept of informed consent existed, Henrietta died never knowing how important her cells would be. Even her family didn't learn of the true extent of her legacy until science writer Rebecca Skloot began working with Henrietta's daughter Deborah to uncover the truth almost 5 decades later.
Okay. 1 second. I am. Yeah. Yeah. So this was about Henrietta Lacks. And so now you know that she was a patient who had cervical cancer. But the researcher, George Gee, was able to multiply her cells. And they are now present all over the world and they're being researched for all kinds of things. And every medicine is first tested on this HeLa cells. They're called HeLa because they're named after her first and last names, the first two letters of her first and last names, HeLa. So these HeLa cells are very important for biological research and they've played a very important role. So this was a short introduction, a brief introduction to Henrietta Lacks. Okay, moving on to what is allometry. So to give you a brief about allometry, it is the study of biological scaling, in which you look at how the size of a living organism is affecting its shape, its anatomy, its behavior, or its physiology. So, any of the characteristics of the living organism and how they're being scaled or they're being affected by the size of the living organism. So you could be looking at morphological traits, for example, relationship between the size of the brain and the size of the body of adult humans. You could be looking at physiological traits, for example, the relationship between metabolic rate and body size among organisms. Or you could be looking at ecological traits, for example, how the wing size and the flight performance in birds are related. So how increasing or decreasing the wing size affects the flight performance. So this is what is allometry, basically studying characteristics of organisms and how they change with the size of that organism. Here is a graph showing. Okay, this is the wrong title. Here is a graph basically looking at how organ size is scaling with the body size. And now you can say this is a graph which involves log x-axis and a log y-axis. So in this you look at how the brain and the heart uh, the size of the brain and the heart get scaled with the increasing body size. So you can see here that heart roughly keeps on growing with the body size. So from birth till like the age of 20, heart is growing linearly with the body size. But when you look at the brain size, much of the brain is developed in its size by the age 5, and after that it's just a flat line. So the brain size does not keep increasing with the body size, unlike the heart. So this is an example of allometry. And there's another thing, which is the Kleiber's law, um, in which you see how the metabolic rate is being scaled with body size in the animal world. So here you can see that this also follows the log scale, and uh, it follows a linear trend again, that the metabolic rate increases with the increase in body size. And this graph follows the equation of y is equal to a into x to the power k, where k here is 0.75. That's like Kleiber has found that value from its calculations, so that's a value that we know of now. So metabolic rate is being scaled along with the body mass. And if you look at the log equation, it is log y is equal to k log x plus log a. Here, k is the slope of this line, y is the value on the y axis, x is the value on the x axis, and log a is the intercept. Okay, moving on to how experiment and theory are different from each other. So, first of all, let's talk about what is theory. So, what do we mean when we talk about a theory in any field of science? So, mostly when there is considerable evidence in support of a general principle and it can be used to explain certain phenomena, then it is called a theory. So, if you do not have a considerable evidence in support to a particular, statement, that's mostly a hypothesis. But when a hypothesis has been experimented and now you have evidence to support that hypothesis, then it can become a theory. In other words, a theory is an organization of facts and data, and the organization of facts and data into a framework, and this can now be used to explain the world. It can be used to make predictions about systems which have not already been a part of the data collection or which have not already been captured in the data collection that was used to come up with the theory in the first place. So, giving you an example of biological theory, as you might already be aware of it, it was the theory of evolution, which is mostly proposed by Charles Darwin, and Alfred Wallace also reached the same theory. In this case, in case of a biological theory, the observations they come from living organisms. So here the observations were coming from living organisms, and in that time the researchers they were trying to classify the organisms, and they were trying to build relationships between these organisms, that how are the organisms related to each other and how can we classify them, or can we come up with one single classification for all of these organisms and to capture all of this diversity. So then Darwin used all the data that he could collect, for example, based on his Darwin's finches, and then he came up with this framework that explained this diversity of living organisms. And he could capture the entire diversity of living organisms by using his theory of evolution and by saying how all every organism is derived from one single ancestral cell, which is called Luca, and then over time, by the effects of natural selection, evolution has come about, and how all of us are related to each other can also be seen in the tree of life. Moving on to how do principles of physics impact the living world? So, when we mostly think about these different sciences, physics, chemistry, biology, a lot of the students might think that they are independent of each other. But that is not true. All of these um, different science subjects have an interplay between themselves. And since physics is the most fundamental science, which impacts all of the objects in this on this world. That is why biology is also constrained by physical principles. Physics and chemistry, both of them have the principles of physics and chemistry. They operate on objects independent of the fact whether that object is living or not. If there is a physical object that exists, then physical principles apply to that object. And since all of us living organisms are also physical objects, we are also constrained in some sense by these physical principles. Okay. For example, if you look at the shape of different organisms, that is also something that cannot go out of the physical principles. If something is not allowed by physics, that will not happen, even in biology. And if we look at how biological theories are different from others, so in essence, biological theories, the answer to this question is that they're not very different, in the sense that they also come about from collecting data, and they also try to explain the different phenomena. All of these theories, as discussed, are constructed by organizing data and looking at the patterns and then trying to predict what is going to happen in some other systems. Biological theories are different only in the sense that the data used to construct them comes from biological objects and not just any other physical objects. So the objects that are being studied, or the subjects being studied in our for building biological theories, need to be living objects. And the mathematical reasoning that can be used in other theories, for example in physics, that is not applicable to the biological world, because biological world is very complex and there are so many variables that cannot really be directly measured. So that is why the mathematical reasoning, which is applicable in other areas of science like physics, are a lot of the times are not directly applicable in biology. Now, later they discussed in the lectures what is a simulation and how the simulations feed into theories. So, simulation is often confused with being a theory, but that is actually not true. And what happens in research is that we begin with a model. And to explain what a model is, it's an abstraction of the real world. So, researchers, what they're trying to do is that they're mostly trying to capture the real world or the reality of what is happening. And they want to do it very close to the reality or very close to perfection. But they cannot capture all of the variables out there because of, say, constraints in a lot of things. Some of the things cannot be measured at all, and some of the things, to measure them, it will require a lot of money or a lot of manpower. So that is why they try to capture reality as close as possible, but by using only a few important variables and not going out and measuring all of these variables. So when they come up with such an abstraction of the real world, that's called a model. And by using this model, they try and run simulations by building some algorithms, either that can be on the computer or that can be by working on a real life biophysical model. For example, was the case with when Watson and Crick figured out the DNA, or the structure of the DNA. They had an actual biophysical model which they were trying to simulate into different ways of how of arrangement and seeing how the DNA molecule is assembled. So that was when they worked with the biophysical model. But now, most of the time, researchers end up working on the computer and on the computer simulations, because there are a lot of these variables that can be very easily tweaked in when it comes to running an algorithm or running a computer simulation, but cannot really be tweaked in experiments. And that is why simulations are often used to replace the experiments. And eventually, if the simulation is giving you correct results or your desirable results, you might move on to uh, perform experiments to verify it further. And whether or not a simulation is correct, or if whether or not a simulation is accurate or successful, that depends on the level of accuracy that is required to answer your question. So it is very important to know what question you're trying to answer, because some of the questions will only need a very coarse scale of refinement, but others might be looking at very close, or very minute scales of measurements. And for that, the accuracy of the simulation might be required, like it might need to be much more accurate than to answer other questions that just answer broad level questions. So an experiment that was talked about during this lecture was the Luria Delbruck experiment. And I have another video here to explain you, which very nicely explains the Luria Delbruck experiment along with some animations. So we'll come to that video in the next slide. Let me just tell you that what happened here was Luria and Delbruck, or Luria mostly, he observed that if you start with a pure culture of bacteria, and you add a bacteriophage, like a virus, then it will clear out the culture, all the bacteria, and it will die. But after a few days or after a few hours, the culture will be back, even if nothing was added to it. So he asked the question, where did this resistant bacteria come from? And then he had these two hypotheses. Hypothesis number one was the spontaneous mutation hypothesis. In this, he said that there was a finite and small probability, there is a small probability that a mutation happened from susceptible virus to resistant, from susceptible bacteria to bacteriophage resistant bacteria, and this will be occurring at a constant rate. So at a constant rate, there is always a mutation happening from susceptible bacteria to bacteriophage resistant bacteria. That is the spontaneous mutation hypothesis. The other hypothesis was called the physiological resistance or induced hereditary hypothesis, in which he said that there is a small probability, a finite small probability, that each cell in the culture might survive the attack of the virus, and all the subsequent progeny from the survival that will now be resistant to that virus. So, in this, what basically he's saying is, in the first hypothesis, there is a probability that anytime, even if the bacteria is exposed to the virus or not exposed to the virus, there is a small probability of a mutation happening, making susceptible bacteria into resistant bacteria. In the second hypothesis, they are saying that the bacteria need to be exposed to the viruses before they become resistant to the viruses. So they cannot. So there is not like a continuous mutation from susceptible to resistant, but they need to be exposed to the virus to become resistant to the virus. So this was the observation, and these were the two hypotheses. So now let's look at what Luria and Delbruck did in order to answer these questions. 1 second. Yeah.
The simple experimental approach that Luria took is to take a bacteria cell and to grow a population of bacteria that are descendant from that cell in liquid culture, so that you go from one cell to billions all in a test tube full of liquid, and then to spread those cells out onto a petri dish that contained the virus T1. Normally, if you put a lot of bacteria on the surface of a petri dish which contains food for them, they will proliferate on the plate. So you'll see lots of bacteria growing all over the surface of your plate. And what happens, though, if you coat that plate with this virus T1, almost all the bacteria are killed, as I said. So you actually see, in contrast to when you have no T1, there's actually almost no growth of bacterium on the plate. But in a few places where a cell is resistant to this virus, what will happen is that cell will now proliferate, generating millions and billions of progeny, which will form a mound of bacterial cells that you can actually see as what's called a colony, a round mound of cells, which all derive from the original cell that one didn't get killed by the virus. So in the induced model, the prediction was that all the cultures would have, let's say, anywhere from 1 to 5 resistant cells in them. While in the spontaneous model, he realized that some of the cultures might have 0, some might have 1, and some might have 50. So why did Luria think that these distributions would be so different? And the reason he thought they would be very different is because, in the induced model, it's the simplest one to start with, all the bacterium that one is going to spread on the surface of the plate prior to ever seeing T1 are going to be sensitive to the virus because they've never seen the virus, so they can't adapt their genetic material in the absence of ever seeing T1. So what one expects to see is, on the surface of the plate, rare cells that have responded to the virus that have acquired the trait. So if one did a series of plates, one would expect that those rare events would happen sort of at similar rates on the different plates. So each plate would have the same number of mounds of cells or colonies. So if it was, let's just give an example that it's 1 in a million chance, and you put 10 plates, each which have 10 million bacterium on them, then you would expect, on average, 10 of the bacteria to expose to become resistant to the virus. Now, if you, in the spontaneous model, where the mutations are happening all the time, and one can imagine that these bacteria are growing up, They're having mistakes in their DNA or in the chemistry that's leading to the mistakes to their genetic material. Some of those changes are going to cause the bacteria to maybe grow a little faster. Some are going to make them be a little bit different in shape, but there's going to be occasional genetic changes that are going to make them resistant to the virus. And that can happen at any time during the growth of this culture. So let's imagine that it happens when there are only 100,000 cells in the culture. The culture has to go through a series of the cells in the culture have to go through more divisions in order to generate millions of cells. And that particular bacterium that's become resistant it has a chance to make more of itself. And it'll make 10 bacterium that are now resistant. And so the fact that the spontaneous change can happen at any time during the growth of the culture and is not confined to when it actually sees the virus, you can see how you could get, in fact, many more resistant cells in a culture than you might expect otherwise. So Lurie's results that the distributions were of resistant cells in the cultures was very skewed and not consistent amongst all the cultures. Ruled out the induced model. It was consistent with the results for the spontaneous model, but in and of itself didn't predict the distributions as Luria originally figured out. He just said that they should be very, very skewed. And so he called in his friend Max Delbruck to help him with the math to say, can we develop a mathematical formula that will actually, assuming there's spontaneous mutations, that will actually give rise to the skewed distributions that we actually see. So it was taking it from being just consistent to, in fact, no, we can actually predict that the results are actually consistent, that they actually show that this is happening by spontaneous mutations.
Okay. So I guess the Luria-Delbruck experiment is now clear to you. So it was a very interesting experiment in which they found out that there is actually a spontaneous mutation happening from sp- susceptible bacteria to resistant bacteria, even without the presence of the virus. One second. Mary? Yeah. So this was all from the lecture in which they discussed experimentation versus theory. And coming to the next lecture, now we're looking at how to find reliable information when you would want to either you need information for your research, or you have been asked as an assignment to write out some article or some project assignment for your course. So one basic thing that everybody does these days is that you can use Google search engine, but you need to be careful while using that information. So in order to make sure that it is also scientifically accurate, you can use specific keywords to refine your search when you're searching for something in Google. Another thing you can do is you can look it up in Wikipedia, but that's like a beginner source of information. And if you want to find more scientific articles, then you should go to Google Scholar. Uh, Give me a second. Yeah. So this is what Google Scholar looks like. And say, for example, you want to look at climate change and its impact on birds, then you can Google that in the Scholar. And here you can see that they tell you a timeline in from which when these articles are published. These are mostly all scientific articles in some books. They will tell you if there's a PDF available of these, you can directly go to the PDF. It tells you it gives you an option to save these. It tells you how many people have cited this article. It gives you an option to cite this article. So if you read the article and you want to use information from the article, you can cite the article in these different formats. And it tells you some related articles of this, and it tells you uh, that it's whether it's available on Web of Science or not, and what's the score there. And it gives you a timeline. It helps you sort by date or by relevance, and you can also look at which articles you want to look at, either research articles, other articles, or review articles. So this is a little bit about Google Scholar, and it is a very helpful website which helps you find scientifically accurate information. You can also use sources like PubMed and bioRxiv. PubMed is mostly applicable to people who are looking at uh, more medicine related research. So if you're interested in things that are related to medicine or diseases, things like that, then you can go to PubMed and find information about that. But bioarchive is another website which has a lot of preprints. It's a preprint server for biology. In this, the articles that are being published are not really peer reviewed, as opposed to other journals that have a peer review process before publishing the articles. So if you have an article that you would like to share, if there's a researcher who wants to share their work um, without it being peer reviewed, they can put it up on bioarchive. And here also you can come across different articles, but you should and there are different subject areas, and you can find out which you would want to look at. It caters to different subject areas in biology. So these are some websites in which you can find scientific articles. So, and I told you how you can go about finding information about impact of climate change on birds. And so, like, these are different articles. You can also type this out in bioarchive and come across different articles. And when you go to one particular article, another way of finding information is that after you have reached one article, you can read its introductions and then go through its bibliography, and you can come across more articles that have been cited in this particular article. So you can go to the bibliography, you can read all of these titles, and from here also you can find relevant articles. And since the date of the publication is mentioned, you can find more recent or more old articles depending on what your project or research is about. Okay, so this is one way of finding reliable information. After you've done that, let's talk a little bit about plagiarism and how to avoid plagiarism. Plagiarism is, as Google defines it, it's the practice of taking someone else's work or someone else's ideas and passing them off as one's own, passing them off as your own work or your own ideas. And there are different types of plagiarisms. One could be a direct plagiarism, in which you're directly copying somebody's work. You're not even changing the words, and you're not giving them any credit. So, that is a direct form of plagiarism. Failing to credit is that you might be taking information and you might be taking ideas from somebody else's work, and you might not be giving them the due credit. So, that is a form of failing to credit somebody's or acknowledging somebody else's ideas and their work. Then there is something called mosaic plagiarism, in which you might actually be paraphrasing somebody else's work, but you're still not acknowledging them. You're still not acknowledging that your source of information, this is not your own information, but you have read it somewhere else. So it results in a mix of original and copied parts. And then there is auto plagiarism or self plagiarism, in which you might be passing off your already published data as a new or already published work as again a new work. So that is self plagiarism. These are some different forms of plagiarism, and it is very important for students to get aware of what plagiarism is and how they should be avoiding it, because a lot of the students are not actually aware of what is plagiarism. They, if they are told to write an assignment, they might directly be copying it off. And these days, since there is chat gpt, you can actually just give it a you can give it a prompt to write you an assignment, and it'll write you an assignment. But that is not the correct way of doing it, and you will not be learning anything by practicing things like that. So therefore, credit where credit is due. You should avoid plagiarism at all costs, and you should go through articles if you're writing some project or some assignment for some course. So, how to avoid plagiarism is that, first of all, just avoid copying and pasting sentences altogether. If you are reading something, if you like some information, always write your own thoughts about it, or you paraphrase it in your own language. Do not copy their sentences. As I said, rewrite information in your own words. You should always cite your sources, where you got that information from, which article you read it from, or which website you got that information from. And if you do want to copy some sentences, say there is something that is so well put that you do not want to change the sentence structure at all, please put them in quotation marks and then cite the source. So when you put something in quotation marks, that means that you've directly taken the sentence from somebody else's work, and then you cite the source where you took that sentence from. For example, you might not be wanting to paraphrase definitions because you like the way it is being defined in that particular article exactly. So you can just put quotation marks and cite the source. Even and important thing to note is that even if you're using images, figures, or videos that are not your own, you should make it a point to always cite where you got that image from, where you got that figure from. And as I said, with the increased use of ChatGPT, increased use of AI, it is very easy for students to generate articles and assignments. But it is important to acknowledge that you have used AI while submitting your assignments. And since most of the times you will not be allowed to use AI, so you should make it a point to actually go through the articles or read up on information before writing an assignment, because that is how you learn something out of it also. Okay. Next thing, some things to make sure you ask yourself. So, when you're trying to avoid plagiarism, if you're writing some manuscript, make sure to ask yourself, are these your own words? Are they your own thoughts, your own ideas? If not, cite the source from where you got them. Did you copy the sentence or the sentence structure? Even if you've copied like a sentence, or the way you're structuring your sentences is very similar to someone else, make sure to ask yourself that and rewrite the sentence in your own words. And when you are citing a source, also make sure that what they are saying is coming across exactly what you are saying also. So, or what you are saying is aligns with what the original or the what your source is saying, because a lot of the times even researchers fail to do this. When you go through literature or the history of some definitions, you find that when it was originally defined, the definition was very different, and over the years, it has changed because of how people have been structuring their sentences. So, whenever you're trying to paraphrase information, please make sure that the meaning is not lost in translation. And next thing is to make sure that you cited the original source properly. Now, since I've mentioned so many times citing information, let's talk about what exactly is citing some information and how do you do that. So, there are different ways and formats in which you can cite some information. One of them is called in text citing. And for every in text citation, there should be a corresponding reference in your reference list in the end of the article. And I would suggest you to follow one same format throughout the document if you're citing articles. And to simplify the process of citations, you can use apps such as Zotero, in which you can make a folder of all of your sources from which you're collecting information and link it up to, say, your Word or your Markdown editor. And then they will directly get cited from just by typing out what you're trying to cite. You will not have to write that entire format on your own. They will directly be taken up from Zotero, all the information. Okay. Now, since I've already talked how to find reliable information and what is a scientific, so it is very important to know what is a scientific article. Since we have mentioned, you know, that you get reliable information from scientific articles. So, what are they? So, there are different types of science articles. One of them could be a research article. In a research article, what mostly happens is that is it involves that the researcher has done some experimentation. The researcher had probably some hypotheses. He or she carried out experimentation and collected some data, and they have their findings, and they want to report these findings to or make it available to their peers or to everybody. And that is when they publish a research article in which they talk about that one particular experiment and what they got out of that. You might be going or reading a review article. In a review article, what mostly happens is that an author summarizes the research articles in that particular subdomain. For example, an author has a question of one particular area, and they want to now summarize all the research that has been carried out in that area. So they will go and they will gather all the research papers from that area, spanning from the earliest papers to the latest ones. And then they might summarize all that research, what has happened, how people have worked out in the field, and what are some research gaps that remain to be addressed in that field. And then there are commentary articles. In these, the authors, they might be commenting on somebody else's research articles. So what they felt about the research articles, what was lacking, and what was you know what was properly carried out and what was not properly carried out in those experiments. So these are some different types of scientific articles. And then if you look at how does an article get published, like how does a research article even get published? So as I said, first of all, the researchers, they might be conducting experiments, and they have their findings, their own theories and ideas, and they want to convey these to the general public or to their own academic circle. So they will write down a manuscript. Then they will submit this manuscript to a journal which is appropriate to their own field. For example, if I'm an ecologist or if I'm an ornithologist, I would want to submit my papers to things like Animal Behavior Society or Ornithological Society, journals of such fields, and not really in journals of, say, physics or medicine or things like that that are not really applicable to my field. Then, most of these journals are peer reviewed, which means that if you submit your manuscript, the editor will then send it out to other researchers who will review your manuscript and see whether or not everything is in place, and if your experiments and your findings seem believable and they are properly carried out. Then the reviewer might they'll go through your articles, and they might now send you comments asking you some clarifications if some things have remained, you know, if they didn't understand some things that you did or how you did them. And then the researchers might be asked to make some revisions. They might be asked to go through their article again and revise some of the statements because their point maybe didn't come across as they wanted it to. And then the editor finally of the journal decides whether or not that paper will get published, either with or without these revisions. Now, once the article has been published, now let's look at what are the parts or what are different sections in a research article. So, when you look at a research article, there are different sections in that article. One is the abstract, which provides you a brief summary. So, an abstract is like a summary of the entire article. What is the system? What are they studying? What did they do? And what did they find out? So, it summarizes the entire thing. Then there's the main section of introduction, which gives you a background in the topic, and they tell you about the prior research, and then they also tell you what their own objectives are in this research paper, and what they are trying to look at. Then they will have a materials and methods section in which they will tell you, in which the researcher will write out what they did and what did they use to do the experiment, and then how they carried out these experiments. So material method section basically is important because science needs to be reproducible. So if I'm writing an article, my materials and method section should be so well written that somebody else who wants to replicate my research should be able to do so easily. And then results will have all the findings that they got from their experiments, and then there will be a discussion section in the end in which the authors will write what they thought about their findings and their own ideas and theories that they want to tell everybody about what they thought of their experiments and what they think is actually happening in the system. So these are the different parts of a research article. If we, for example, say come so we were looking at, so for example, say we are looking at this article which looks at the potential effects of climate change. So as I said, there is this abstract, so you can download the PDF. So in some of these places, they will allow you to download the PDF. But okay, as I said, there will be an abstract section in which they will tell you the brief summary of what is happening in that particular article. Then they will move on to giving you a brief introduction of the background of the research. Then there'll be the materials and methods section in which they tell you what they did in this paper. And from the methods, then they'll come to the results. What were the findings? There'll be some figures that outline the findings that they got, the statistical tests that they ran for this paper, and then there'll be a discussion or the conclusion section in which they talk about what is happening in these areas. Okay, so these are some parts of a research article. Then when you talk about how to read research papers, so when students start out with looking at research papers and reading research papers, it's not very easy. It requires a lot of patience, and these research papers are very information heavy. So if an amateur is starting to read out research papers, it takes a lot of the energy and a lot of concentration, focus, and patience to understand these research papers. So Dr. Shavanti, in her lecture, she gave this brief outline of how she thinks it's helpful for people to start out by reading research papers. She says that you should always start by reading the title and the abstract, so that you get an overall idea of what exactly is happening in this article and if it is even relevant to your research or to your project. So by reading the title and the abstract, you get a rough idea of what is happening in this article. And then if you're a beginner, it might help you to read as a group. So you might have a group of batchmates, or a group of people working on the same project or similar things. So you might want to read as a group so that you can discuss the paper and you can clarify the doubts with each other. Then, after reading the title and abstract, she said you should skip the introduction and then move on straight to the material and method section to see what the researcher actually did in this experiment. And side by side, you should start making a term glossary, because a lot of the times, as I said, these research papers are information heavy. They will have a lot of jargon which is applicable to their own field, and you might not be aware of it. So it will be helpful to create a glossary for yourself. And then you should, after reading the material section, you should move on to figures and their captions, and not read the result section, but look at the figures, because from the figures you'll get an idea of what their findings really were. And now, when you've done this, when you've read the title, when you've read the abstract materials, and then you've read the methods, and then you read the figures and their captions, then you should move on to read the entire paper, because now you already have an idea of what they did and what they find out. So it will be easier for you to now go through the paper and understand what they're trying to talk about. So this is Dr. Shavanti's view on how to read research papers. But very ironically, there are even research papers that talk about how you should go about reading research papers. So if you're really curious about how I should start my journey of reading research papers, you might want to read some papers that talk about how to read papers. And if you still don't know where to start, then this I Think Biology book itself, or the website I Think Biology, that is one of the resources for this course, has these articles that are very nicely annotated for your use. So, for example, there's this article which talks about leopards providing public health benefits in Mumbai, India. So, this is the title. Here is the abstract. And here, a lot of the lines are underlined and a lot of the meanings are written. There's a glossary given, so it'll be easier for you to read papers that are provided already in the I Think Biology website. That might be a good starting point to know how to understand papers. So this is the last slide, in which we talk about how do you summarize a research paper. For example, your assignment was to summarize some paper, or you want to give a presentation about some article that you read. So for summarizing the paper, in her lecture, Dr. Shavanti said that you should summarize, first of all, start by giving an introduction. So, again, you also provide a background information to give some context of what was happening and and why was this an important experiment? What did they find out? What is the history behind it? What is the background? What is the system? You introduce the system of, and then you tell what is happening and what they're trying to look at. So that is an introduction that you should provide. Then you should give some subheadings. You should use figures as the guidelines, the figures that were present in the results section and in the other sections, and use them as guidelines in order to discuss what is being done in the paper. And then you move on to the conclusion and discussion section, and there um, you write in your own words what you thought of the paper and what is being addressed, what was the major question, what did they do, and did they cover all the necessary control experiments, why are these results important, and all these things. And last but very important is that you provide citations to your information as well. What you're writing out, you also cite whatever information you got and where all you got it from. So that is how you can go about summarizing a paper. Moving on to the question and answer section for today. So as this is a problem solving session in which we mostly in the end we have a Q&A section in which we look at some MCQ questions, so that you know what to expect from the assignment and from the exam of this course. So the first question for today is, you can either type your answers in the chat box or you can unmute yourself and tell me the answers. The first question is, a good theory must have testable predictions. Is this true or false? Should a good theory have or not have testable predictions? So, is this statement true or false that a good theory must have testable predictions? Correct. This is true. As we've already discussed, science is falsifiable. That is one of the central themes or central ideas of science. So, therefore, a good theory must have predictions that can be tested and uh, falsified. In the discussion with Dr. Shashi Tuttapalli, he described the Luria-Delbruck experiment. This experiment tested what? Did this experiment test uh, whether bacteria can survive a viral infection or if bacteria have mutations? Mutations that arise spontaneously in bacteria and not in response to viral infection. And viruses and bacteria are symbiotic in nature. So, which of these statements describes what was being tested in the Luria-Delbruck experiment? Any guesses? We just saw a video about the Luria-Delbruck experiment in which, and before that, I also gave a brief about this experiment. So basically, what Luria and Delbruck were trying to do was to see if mutations can arise spontaneously in bacteria and not really in response to a viral infection. So that is what they were looking at during this experiment. Ki whether mutations arise spontaneously or do they only arise in response to a viral infection. Next is this image below shows relative sizes of different organisms on the logarithmic scale. After studying the image, determine how many viruses will need to join from end to end to make it equivalent to a plant cell. So, what they're asking in this is, so in this you have viruses and how many viruses need to join to make it to a plant cell. So, if you look at the size of a virus, here is a virus and the size of a virus is 100 nanometers, which means 100 into 10 to the power minus 9 meters. And what is the cell of a plant cell? The cell of a plant cell is around 10 micrometers, which is 10 into 10 to the power minus 6 meters, right? So, if you want to know how many viruses need to join end to end to make it equivalent to a plant cell, oh, the answer already came on the screen, which is B100, but okay. If you want to look at it like this is how you would estimate it. And here you cancel out this 10 and it becomes 10 to the power minus 7 into 10 to the power 9, which will give you 100. Okay, I hope you understood this calculation. So basically, you divide the cell of the size of the plant cell with the size of the bacterial cell. So 100 or the size of the virus, yeah, B is the correct answer. 100 viruses need to be end to end to form a bacterial cell or plant cell. Next is what is allometry? Allometry refers to the study of the proportional growth of different body parts in relation to body size, classification of organism based on physical traits, study of animal behavior, effect of climate change on ecosystems. So, which of these statements do you think is closest to what allometry is? Okay, so let me tell you the answer. Allometry basically refers to the proportional growth of different body parts in relation to, yeah, in relation to body size. Correct, it's part A. So it's not just the proportional growth of different body parts. In general, allometry refers to how different characteristic of an organism scale in relation to the body size. So it could be any characteristics, not just body parts. But out of these four statements, A is correct. Which of the following is an example of a nominal scale in biology? Body temperature, heart rate, species name, or weight. As we discussed during the class, nominal scale basically is a scale in which you only have classifications, in which you only have category names and not really a scale. You just classify the variables into different, or the values into different categories. So which of these do you think is a nominal scale? It is not really a measurement scale, but just has different category names. Exactly. The answer to this is species name. So, giving different organisms a name or giving different species a name, that is a way of nominal scaling in biology. What type of numerical scale is body length in centimeters? So, which of these is body length in centimeters? Is it a nominal scale, ordinal scale, interval scale, or a ratio scale? So, as we have already, the answer is the ratio scale. As we have already discussed, nominal gets cancelled out because it is just naming different categories and does not have any scaling. Ordinal is just the values following a particular order, for example, a ranking system. So, this also gets cancelled out. Interval is a scale in which the different values have a particular interval, but there is no real zero. So there is no real zero in an interval scale, and ratio scale is the only option which has an actual zero as well. And since we are talking about body length in centimeters, so there is a real zero in centimeters or in body length. That is why it follows a ratio scale. I hope that is clear. Moving on to the next question. What is the defining characteristic of an ordinal scale? I've already talked about this in this previous slide, so you might be able to answer this one better now. What is the defining characteristic of an ordinal scale? The values have no inherent order. The values have an order, but they do not have a fixed interval between them. The values have equal intervals and a true zero. Values represent continuous measurements. So, which of these? Correct. It's the B, in which the values do have a particular order, but they do not have a fixed interval. As I said, it's like ranking students in a class. So they do have a particular order in which first, second, third, but the interval might not be the same between them. So when reading a scientific paper, which section is typically read first to quickly understand the study? Methods, results, abstract, or references? Which section will give you a quick overview of the study? Yeah, since we've already discussed this, it's actually the abstract. Abstract will give you a quick overview of what is that scientific paper about. Moving on, which section of a scientific paper provides background information and the research question? So which of these sections talks about what the system is about and what the question is that they're trying to address in this paper? Abstract, introduction, methods, or discussion? Correct. The answer to this one is the introduction, in which they talk about what is the system and what they're trying to study in that system. Okay, what does the term self plagiarism mean? Is it copying someone else's work without credit, or publishing your same research multiple times as if it was something new, citing your own work too many times, or making up fake citations? What is self plagiarism? Yeah. So again, the correct answer to this is option B. When you're trying to pass off your same research as a new work again and again. Okay. What is the main purpose of the discussion section in a scientific paper? So what is the purpose of the discussion section? Does it describe how the experiment was conducted? Does it summarize the findings and interpret what the significance of the findings? To list the references or to state the research hypotheses? Correct. Again, it's the B option. In the discussion section, they summarize the findings and they're trying to interpret what is the significance of these findings. Which of the following does not require citation in a research paper? A sentence that was copied from another paper, even if rewritten in your own words. A well known fact, such as water boils at 100 degrees Celsius at sea level. A paragraph from a book, or a data point taken from another's research article. So, which of these Yeah, you're correct. So if it's a well-known fact that is that has been known for a long time now, it does not require citation in your paper. Next question: which of the following is an example of plagiarism? So, which of the following is an example of plagiarism? Summarizing an article in your own words and citing the resource, copying and pasting sentences from a research paper without crediting them, using publicly available data with proper citations, or paraphrasing a study and giving proper credit. Which of these is an example of plagiarism? You're correct. It's the option B. If you're copying and pasting sentences and not crediting the author, that is plagiarism. If you summarize someone else's study in your own words, what should you do to avoid plagiarism? Should you do nothing because it's in your own words? Should you be adding quotation marks? Should you be citing the original resource? Or should you be replacing a few words and keep the sentence structure the same? So, what should you do in order to avoid plagiarism if you are summarizing someone else's study in your own words? Correct. You should be citing the original source or the original article where you read the work. Last question for today is: what is the best way to cite an article within a research paper? So, out of these options, what is the best way of citing an article? According to Smith 2020, climate change affects biodiversity, or climate change affects biodiversity, and then you are putting the citation Smith 2020. Both A and B are correct depending on the sentence structure, or citing is unnecessary if the article is well known. Which of these is the correct way of citing uh, an article in a research paper, or are both of them okay depending on how you are structuring your sentences? Yeah, so both of these, A and B, are correct ways of citing the source depending on how you want to write your sentences. So this was all for today. If you have any doubts, you can unmute yourself, or you can type it out in the chat box, and I am happy to answer your doubts. Otherwise, we're done for the day. You can exit the meeting. Thank you for joining, and this was all for today's session. Thank you. Yeah, I will end the meeting then.